One of the most common claims by race realists is that folk racial groups are subspecies. This claim is actually difficult to interpret because subspecies do not have a singular definition. They are also not universally accepted in modern taxonomy and systematics, and most fields do not actively use subspecies concepts. As David Hull said,
"In general, professional systematists do not place much weight on the groupings below the species level. And as Ernst Mayr said in his Of What Use are Subspecies, it became clear that subspecies was not a concept of evolutionary biology, but simply a handle of convenience for the clerical work of the museum curator."
However, four main subspecies concepts have been employed in the past. First,
subspecies are geographically circumscribed groups within a species that have a high level of within-group genetic similarity and a low level of between-group similarity.
Second,
ecological subspecies are local populations within a species that are adapted in important ways to their particular environment.
third,
populationist subspecies are groups within a species that exhibit some noticeable cross-locus genetic differences due to partial genetic isolation.
fourth,
Cladistic subspecies are relatively large groups within a species that form distinct lineages, or, in other words, there have been only a small number of reproductive events that have taken place between these large groups over an extended period of time.
But how do we determine just how well folk races match these different subspecies concepts to gauge the accuracy of the race realist position? The first subspecies concept claims that human races are geographically circumscribed groups within a species that have a high level of within-group genetic similarity and a low level of between-group similarity. Traditionally, this definition has hinged on FST estimates, which was referenced last video. Generally, Lewontin's results and the correspondence of low FST values in later studies like Rosenberg et al. 2002 have been taken as evidence that between-group differences are small, and Edwards' work, as well as results from structure, are taken to be evidence that individuals can be grouped in a racial genetic cluster. However, as noted, Edwards did not challenge Lewontin's claim that the between-group differences were small.
Myth 1. FST values lower than those observed in humans are used to claim subspecies in other species. A common race realist meme is a table of FST values and number of identified subspecies with a cited paper. One major problem with this argument is that the literature actually doesn't support it. Instead of those cited papers using genetic levels to support subspecies classifications, they all apply genetic technology to systems that have previously described subspecies, and then look at the genetic differentiation between those groupings. Obviously, the genetic evidence cannot be used to justify pre-existing categories. They can only describe the difference between the already identified categories, and frequently in these studies, the low genetic differentiation and the lack of isolation between subspecies is used to argue that many subspecies are invalid units for the system. This was the case for zebras, cob antelope, and African buffalo. This follows a pattern in modern systematics and population biology, where subspecies ranks are eliminated and either merged or split to distinct species based on genetic data. In a 2004 study, Ryan Zink used the small amounts of genetic variation in population structure in bird species to challenge nearly 97% of claimed temperate bird subspecies that he investigated, even though many of them showed some level of non-random genetic variation. Primate taxonomy has also experienced a major drop in subspecies and an increase in identified species based on new data and a more robust concept of species that aims to identify independent evolutionary lineages as the smallest unit in taxonomy.
Myth 2. Lewontin's fallacy is real and shows Lewontin was wrong about the insignificance of race. More work has been done since Edwards, and the details of the debate shed more light on Lewontin's actual claims about the significance of racial groupings. One important improvement is that a critical appraisal of FST's appropriateness as a method has been carried out. Jeffrey Long looked at the assumptions of common methods to compute FST to see if they match certain facts about human populations. In their own words, we ultimately found that the genetic and statistical model underlying FST does not fit well to human populations. Specifically, human population structure strongly biases the outcome of analyses by violating two assumptions. First, that expected genetic diversity is the same in every population, and second, the divergence between all pairs of populations is equal and independent. They also find that FST measures are highly dependent on the frequency of the allele being analyzed, which can bias FST values further.
Using this information, Long performed two further analyses that correct for these violated assumptions. The first was to compare FST values between humans and chimpanzees. An older analysis, without model correction, the FST between humans and chimps was 0.18, a modest increase from the 0.12 for humans alone. Using the updated model, the FST between humans and chimps is twice as large as in the older analysis, which better corresponds to our understanding of divergence between humans and chimpanzees and showing how important these model assumptions are to the results of these experiments. More importantly, Long and others formally tested how well genetic diversity data fits different models based on racial taxonomy. The idea is simple. Measure genetic diversity in populations and then predict patterns of genetic differentiation in the same populations using different models representing different racial concepts. Then use statistics to determine how well those models match actual genetic diversity. The model for traditional FST analysis has the worst fit using both single nucleotide polymorphisms and microsatellites, while the adjusted model that allows for hierarchical relationships between subpopulations fits better, although still not perfectly, signifying the complex structure of human populations. The conclusion from this experiment is that if races are supposed to be subpopulations in an FST analysis, or from algorithms-like structure, they do not match patterns of human genetic diversity because they will overestimate diversity out of Africa and underestimate diversity within Africa. This simple race concept, therefore, does not accurately represent human genetic patterns. The other conclusion is that any race one potentially could identify from the hierarchy model using historical race concepts do not match folk races because Africans cannot be a race, since the most exclusive group that includes all Africans also includes all other races. It would also make Eurasians a single race, requiring several sub-races to get European and Asian populations to separate, as we generally think of folk racial groups. Long's work makes a few things very clear. Based on genetic divergence data, folk races do not reflect the actual patterns of human genetic diversity and are not legitimate taxonomic units, and the other races that may exist from human patterns are not folk races as traditionally thought of. The second subspecies concept claims that races are local populations within a species that are adapted in important ways to their particular environment. This subspecies concept, laid out by Massimo Pigliucci and Jonathan Kaplan, uses the ecology concept of ecotypes. However, Pigliucci and Kaplan are quick to point out some key facts of ecotypes.
1. Ecotypes are not necessarily evolutionary lineages. 2. The same ecotype can evolve independently in different groups. 3. Ecotypes will not be discrete units because adaptive variation typically varies continuously in a clinal pattern. 4. Each individual will belong to multiple ecotypes or races, not only those who are usually considered mixed race. 5. There will be many more races according to the ecotype approach than are traditionally presumed to exist.
This would mean that folk races could not be thought of as ecotype races in any simple way. For example, although skin color is adaptive and broadly differs between continents, both some African populations and some Aboriginal Australian and Melanesian-Polynesian populations have equally dark skin based on light reflectance data. Additionally, some African hunter-gatherers have skin as fair as some Asian populations, with new evidence that light skin alleles were selected upon after backflow from an out-of-Africa population about 2,000 years ago. Some traits that people think of as racial traits, like sickle cell anemia and the accompanying allele, is found in similar frequencies in part of Africa, the Arabian Peninsula, India, and the Mediterranean. Likewise, there are several areas in Africa where sickle cell is not found at high frequency. We could continue down the list of traits like lactose tolerance, which is found in both African and European populations, to craniofacial traits which form a geographic cline similar to neutral genetic variation, and we will see a general pattern that no trait can form an ecotype, or no collection of traits can form consistent ecotypes that would produce folk racial categories like white and black. This is also a pattern that clustering algorithms like structure cannot save because genetic population structures based on the neutral loci do not tell us anything about the distinctness of the environments inhabited by the respective populations or how these populations have adapted to them. This disconnect between environmental niches, genes, and geography suggests that we cannot tell whether two groups are distinct ecotypes merely by looking at the genetic and geographical facts. In fact, under this subspecies concept, two populations from the same cluster, Han Chinese and Tibetans, could be different races due to adaptation to high altitudes in Tibetans, but two populations from different clusters, such as Tibetans and indigenous Andean populations, would be the same race. A larger problem with the ecotype subspecies concept that Alan Templeton has pointed out was that focusing only on adaptive traits ignores most of the genome, and that there is no objective way of choosing which adaptive traits to racialize on the ecotype approach. Different adaptive traits display different geographical trends because the underlying environmental factors have different geographic causes. As a result, one will get different ecotypes for different adaptations. Depending upon which adaptive trait is chosen, one will get very different races. So it is impossible to justify one particular ecotype arrangement over the other competing ones. The evidence for folk races being a subspecies does not seem to be well justified with this concept, and the needless arbitrariness of this subspecies concept makes it a very weak category in general. Now onto the third subspecies concept, the claim that races are groups within a species that exhibit some noticeable cross-locis genetic differences due to partial genetic isolation. This would appear to be a subspecies concept most likely to hold, but it is actually the weakest. This concept largely stems from the evolutionary biologist Theodosius Dobzhansky, but even he recognizes the flaws with this race concept. The obvious flaw in such a definition is that differences in gene frequencies may be quantitatively as well as qualitatively of diverse orders. The statement that two populations are racially distinct really conveys very little information regarding the extent of that distinction. If this is truly the kind of race realism that is being defended, it means very little since populations are not highly genetically diverged. Putting this issue of magnitude aside, though we can see if folk races meet this category, many race realists point towards clustering programs like structure for support. This subspecies concept includes partial genetic isolation, which we cannot infer from programs like structure. In fact, identifying whether the source of population clusters from structure is from artifacts of sampling too many or too few individuals from a population, and or from structure's tendency to make clusters based on the fact that there is more gene flow in geographically close populations than in most distant populations, something called isolation by distance, instead of genuine geographic barriers, is a major task for these kinds of studies. Some, like Kalinowski, have shown that clusters generated by structure appear to be due to undersampling Africa and oversampling Europe. And Hanley et al. Showed that of the total genetic differentiation between populations, less than 2% is from discrete clusters, while over 75% is from geographic distance from Africa. These collectively appear to be evidence that there is not actually significant partial genetic difference between folk racial groups, and that, like in the case of rats in Manhattan or European populations, the existence of genetic clusters does not mean the existence of races or subspecies. This should not be taken as a claim that population structure does not exist. Obviously, human populations around the world differ in gene frequency, but rather the nature of that population structure does not lead one to conclude that structured populations are subspecies. There are many questions one can ask about human genetic diversity, and a particular question can be approached with different goals and methodologies, which produce different numbers or arrangements of clusters. However, nothing makes one question or one way of organizing genetic clusters the obviously correct one. The philosopher of biology, Adam Hochman, has convincingly argued that certain conditions need to be met before we move from genetic clusters to races. Otherwise, the race realist position would not differ from the anti-race realist position held by most population geneticists and anthropologists.
There should not be more difference in genetic diversity between one cluster than between that cluster and another. The number of clusters should not be arbitrary. The allele frequencies between a cluster should be relatively homogeneous. And there should be a large jump in genetic differences between clusters.
But none of these conditions are met. 2 Africans can be more genetically different than a European and a Japanese person. As the Manual for Structure says, in the case of real-world datasets structured by isolation by distance or inbreeding,
there may not be a natural answer to what is the correct number of clusters.
And Hanley et al.'s analysis showed that virtually all allele frequency differences are clinal, and there are no large jumps in genetic differences between the clusters. Without the meeting of these conditions, there is no straightforward link to stronger biological racial realism. It is also important to note that Long's analysis that observed patterns of genetic diversity do not match folk racial categories also applies to this kind of race concept, meaning not only can we not produce a positive argument why folk races uniquely meet this kind of subspecies concept, we have evidence explicitly showing us how poorly this race concept accounts for human genetic diversity. We also have evidence that there is no way to distinguish which racial groupings is the best or the correct one. And finally, the 4th kind of subspecies concept says that races are relatively large groups within a subspecies that form distinct lineages. Or, in other words, there have been only a small number of reproductive events that have taken place between these large groups over an extended period of time. This is one of the most clearly defined and testable race concepts, but subsequently it is one of the most disproven race concepts. The claim is simple. One can produce an evolutionary tree based on the genetic loci or genetic distances, and that tree represents populations as distinct evolutionary lineages branching off from each other. But the conclusion is not so simply made. The problem is that one can frequently represent a population using a branch diagram even if the population does not actually contain any isolated breeding populations. As the evolutionary biologist Alan Templeton puts it,
the computer programs used to generate trees from genetic distance data will do so regardless of what evolutionary factors generated the distances. After biologists make trees based on the genetic distances between the populations, they do the further step of testing whether the populations do in fact fit a tree-like structure or treeness. And, Templeton points out, the failure of human genetic distances to fit treeness is ubiquitous whenever tested. Not one of the datasets fits treeness.
Templeton also used an analysis called multi-locis nested clade phylogeographic analysis, which is a mouthful, but all you need to know is that it can look at the evolutionary pattern of multiple loci to identify when populations split and merged throughout their history. Multiple evolutionary trees were made for human populations and tested in a statistical framework, whether to see if any of the population splits have occurred. This would mean that a subset of the species splits off from the remainder of the species and behaves primarily as an isolated lineage of race after the split, even if some limited gene flow or introgression still occurs. It can also test the null hypothesis, that populations from 2 geographic regions have experienced no gene flow in a specified time period. This method firmly rejected any splitting or fracturing of human lineages, meaning that genetically, humans have never stopped being a single evolutionary lineage and supports the continual gene flow and admixture among all populations from 1.9 million years ago to the present. For this type of race concept, there is no evidence that humans have or had races at all, whether they were folk races or some other possible groupings that may or may not reflect folk races. In closing, I'd very much like to thank Kevin Bird for his assistance on this project. He has forgotten more than I know, and I would have been absolutely lost without him. Check him out on Twitter because, in addition to the time he lends you should you ask, he's also a really great guy. He's always given me a fair shake, and I must close noting how appreciative I am of that. This series could not have happened without you, Kevin. I'd further also like to thank the many kind people who have lended their voices to this project. Claudia Brown, Crownton T, SJW Debates, Pig Puncher, and Legend of Phoenix. Links to their respective content will be left in the description. They are all wonderful people. Thank you.